Hi everybody, today we are going to talk about how the cell cycle is regulated. So our topic today is regulation of cell division. And we now know about mitosis, we know how mitosis works, we know how the cell cycle works, and now we're going to find out how does a cell regulate when it divides and how quickly it divides. So coordination of cell division, it says here that multicellular organism needs to coordinate division across tissues and across different organs because they all have their own specialized functions. So normal growth and development and normal maintenance is going to require some communication and some signaling. So timing is going to be really important, and the rates of cell division will be really important because not every cell type has the same timing for their cell cycle. So let's take a look at how the frequency of cell division may vary. So first of all, we have an embryo. So an embryo is a rapidly growing animal. And so the cell cycle is going to happen very fast because embryos are growing very quickly. The cell cycle has to happen very fast. So each cycle through interphase, through mitosis is going to take less than 20 minutes. Skin cells are constantly dying and being replaced, so they are going to be replaced pretty quickly as well. Every 12 to 24 hours, your skin cells are going to go through the cycle and reproduce. Your liver cells kind of keep their ability to divide reserved, but they might divide every year or two, so they divide pretty slowly. And some cells, like mature nerve and muscle cells, will never divide because they are permanently in G0. So there are two irreversible points in the cell cycle. The first irreversible point is when the genetic material has been replicated. So once you get there, once you've replicated your DNA, the DNA is replicated. You can't go back. So that's what we see here, right? We see a homologous pair that is unreplicated becoming a replicated homologous pair. All right. We have the separation of sister chromatids, as you can see in the picture here. Once we go through mitosis, each duplicated chromosome is going to separate like this. Once they're separated, that is also irreversible. You can't go back and put them back together. There's no enzymes for that in the cell. Checkpoints throughout the cell cycle that is going to assess these processes and make sure everything is happening normal. And if things are not happening normally, then the process gets halted. All right, so we have a checkpoint. Did duplication go okay? And then we have another checkpoint over here on the right. Did separation go okay? All right, so here is the main control system. We have checkpoints. The cell cycle is controlled by various stop and go chemical signals. And we have this analogy here. We have a checkpoint in the G1 towards the end. We have a checkpoint after G2, and then we have a checkpoint midway through the M phase. So these are actually chemical checkpoints that keep us from moving on. The signals indicate if key cellular processes have been completed correctly. And let's learn about these three major checkpoints. So first of all, there's G1. G1 happens, so this is the beginning of the cell cycle. Follow my highlighter. G1 begins, and towards the end of G1, we've got this checkpoint. Can we move on to the S phase or not? Can DNA synthesis begin or won't it? So questions that the cell will have to answer: is the cell big enough? Are there enough nutrients? And do we have signals from other nearby cells that there's enough room and enough surfaces for the cells to grow? So those are the questions asked during G1. If the answer to those is yes, then the chemical signal will be received and the cell will move on into S phase. G2 checkpoint, which happens at the end of G2 after the DNA has replicated. So has DNA synthesis been completed correctly? If there are major issues with DNA replication, then the G2 checkpoint is not going to let anything through. Once we go through the G2 checkpoint, we are committed to mitosis. It is happening. The cell is dividing. And then we have our metaphase checkpoint. That is also called the spindle checkpoint. And there we are making that the chromosomes have attached on each side to a spindle fiber. Because what's going to happen if the chromosomes are not attached to a spindle fiber? Well, if they're not attached, they're not going to pull apart correctly, and you're going to end up with a wrong number of chromosomes. So this is an important checkpoint as well. Can the sister chromatids separate correctly? That is the question the cell has to answer. So we have all of these checkpoints. So we have a stop here. This is the main stop. If everything is a go, then imagine like a traffic arm going up, allowing us to continue. DNA replicates. So at the end of G2, we're checking, did DNA replicate properly? If it did, traffic arms going up, we're able to go through, mitosis begins, and here we have our metaphase checkpoint. Is everything okay? Are all of those chromosomes attached to a spindle fiber? And if so, the cell goes ahead and divides, and the whole thing begins again. So 3 important checkpoints. There might be more, but these are the 3 main ones. The G1 checkpoint is the most critical checkpoint. This is the primary decision point because this is the main restriction point of a cell dividing. This is the main thing that's going to stop cell from moving on and dividing. Typically, if a cell receives a go signal here, everything else is going to go normally. So this is the main way that things can go wrong. What we need to make sure the cell is big enough to divide. And we need to make sure the cell has the appropriate nutrition. It's got to have plenty of energy if it wants to divide. We also need external factors. So things from other cells, signals that are letting the cell know that it's okay to grow. Cells need room. If there is not room, if the other cells are telling it there's no empty space, then those growth factors are not going to be signals that allow the checkpoint to move on. So if the cell does not receive these internal signals and these external signals, it's going to exit the cycle and it's going to pause in G1, so it will be in G0 phase. Sometimes a cell can be returned back to G1. Again, this is review. G0 is a non-dividing differentiated state. Most human cells that are in the G0 phase include your liver, muscle, and nerve cells. Okay, how do cells know when to divide? Well, it's chemical signals like we talked about in the last chapter. It's signaling that tells us if a cell can divide. Here we have a cell in the M phase, so it's in the process of dividing, and here we have a cell in the G1 phase, so mitosis, and here we have a cell in interphase. Well, what has happened in this experiment is there are chemical signals that are making the cell undergo mitosis, and those chemical signals are absent in the cell on the right. When we fuse these 2 cells together, now those chemical signals can flow between them. So what's it going to do to the cell on the right? It's going to tell it start undergoing mitosis. And so you see here that the cell formed a chromosome. Now notice the chromosome isn't duplicated because we were in G1 and the chemical signal told it to immediately go into mitosis. So it didn't get the signal to divide its DNA. It got the signal to form a chromosome. So these are chemical signals giving it a cue. It's timing by chemicals. And so this experiment is evidence that it's chemical signals usually proteins, which are usually controlled by activator inhibitors. And there is the experimental evidence. All right, so what are the go-ahead signals that we can use to push a cell forward in the cell cycle? Well, there are protein signals that are going to promote growth and division. Internal signals called promoting factors. And then there are signals called growth factors. We usually need both to push a cell past that G1 checkpoint. And this is the mechanism of control is going to be phosphorylation. We see here we have a cyclin-dependent kinase. This is a protein that has been phosphorylated, and that's usually going to activate a protein. So kinases are going to do phosphorylation, and phosphorylation can activate. Sometimes it can inactivate a signal, though. Okay, so what are some of these signals that need to be received? there are chemicals called cyclins. A cyclin is a regulatory protein that has levels that go up and down depending on the phase. We have CDKs, that's a cyclin-dependent kinase. So this is a kinase, it phosphorylates something. And it is going to phosphorylate cellular protein, so it's going to cause a signal transduction cascade. And it can activate or inactivate proteins. When these two things come together, like you see in the picture, the CDK and the cyclin, when they come together, they make what is called a mitosis promoting factor, an MPF. And the mitosis promoting factor is going to push the cell past the G1 checkpoint and on into S and G2. So MPF is both of these together. All right. All right, that's an activated CDK. Triggers passage through different stages of the cell cycle. So cyclin-dependent kinases are enzymes that are present in the cytoplasm at all times. We always have some CDKs. CDKs are usually inactive unless a cyclin is bound to them. This is an important point to remember. We have our CDK, it's inactive. It only becomes activated when a cyclin is bound to it. Together, the CDK cyclin complex that's sometimes called a mitosis promoting factor is then going to be activated, and it can start phosphorylating proteins like you see here. And this phosphorylated protein is going to act as a signal that's going to move on down the transduction pathway. All right, the amount of cyclins in the cell is going to change throughout the cell cycle because cyclins cycle, they get degraded and then they get synthesized again throughout the cell cycle. So basically, when we don't have cyclins, we don't have activated CDK, and when we do have cyclin, we do have activated CDK. So if we start at the beginning, we start in G1, we have CDK, it's inactive, and over time, we build up more cyclin. As more cyclin builds up, it interacts with more CDK to push us through to the next phase. And then the same thing happens. We have other CDKs, more cyclin is made. This is cyclin for the S phase, and it's going to push us through that checkpoint. And then it happens again for the metaphase checkpoint. So we have these levels where cyclins build up, and then they get degraded after the checkpoint is over. So they build up before the checkpoint if everything is normal, and then they get degraded after the checkpoint. All right, so here again, CDK present. When it's time for the checkpoint, cyclin starts accumulating. So there's plenty of CDK. It's inactive though. Now we have more cyclin being built. The cyclin is going to produce MPF that will push us through the checkpoint. After we get past the checkpoint, the cyclin gets degraded and the whole process starts again. Basically, we always have CDK, but cyclin will only build up if everything is normal in the cell. We can only make cyclin if everything is okay. So if the cell didn't, in this case, undergo the S phase correctly, if DNA replication didn't happen the way that it should, are we going to make cyclin? No, we're not going to get the appropriate signals to produce cyclin. So this wouldn't happen. This will only happen because this is the G2 checkpoint. It will only happen if DNA replication occurred the way that it should. So, synthesis of cyclin begins through the S phase and continues, assuming everything is normal. The cyclin combines with the CDK to produce some MPF. The MPF is going to then phosphorylate other proteins, pushing the cell through to mitosis. Then the cyclin is going to get degraded, and the CDK will be alone. Here we have a graph showing the activity of cyclin. Cyclin goes up. When it peaks, we have peak MPF activity, and we push the cell through to the next phase, and then the cyclin goes down, so the MPF is no longer activated and it goes down. And at the same time, in the next cycle, again, cyclin builds up, MPF activity goes up, we get pushed through to the next phase, everything goes down, over and over and over again. All right, CDKs and cyclin drive the cell from one phase to the next. Proper regulation of the cell cycle is key so that the genes for these regulatory proteins have been highly conserved. We've talked about this before, highly conserved. What does that mean? Highly conserved means that it hasn't changed much over time. This means that us and things that are very unrelated for us have similar mitosis promoting factors. This means that they are very effective in doing their job. Evolution has kept them around. So that's an important point to remember. Right? The genes are basically the same in yeast, insects, plants, and animals. The genes for cyclins in us and the genes for cyclins in yeast are very similar. So that means they're highly conserved. All right, we are going to talk about external signals another time. So we're going to stop here. Let me know if you have any questions.